I'm delighted to introduce Dr. Rosa Xicola for today's speaker. So Rosa actually, right now is the first year of our laboratory genomics fellow. She definitely has a special pathway come to here today. So since the first time, I'm going to do a very brief introduction for her training and experience. She received her BA and PhD from Universitat Autónoma de Barcelona. That's probably all the close I can get a Spanish version for the column. Then she came to the States and did a postdoc. She actually went to the England first, very short, and then came to the States, did a postdoc in the Chicago University of Illinois, and then came to the Yale, mostly in the digestive disease section, which probably dedicated most of her research in the cancer-related research, and actually could promote it as AIS, and then in 2017, she was promoted as an assistant professor in the digestive disease section, which I mentioned earlier. She has a special sort of a pathway, and then she became a fellow last summer in our DNA diagnostic lab. So she published quite extensively at a short career here, published more than 60 papers and a chapter for the book chapter, and then also received the award from 2017 Kirshland Junior Faculty Award from Digestive, I guess that's from the Department of Medicine at Yale School of Medicine. And she has been in our lab for about 7-8 months. She has been very active to learning, of course, to contribute to our DNA diagnostic lab from her knowledge experience. So she has a very interesting title today, so I'm very looking forward to hear her talk. Rosa, the floor is yours.
Perfect. Thank you for the introduction. So I'm going to share my screen. Okay. So my talk today is entitled miRNAs and Genetic Disorders. Oops, that's another one. Okay, so the talk objective today is to understand microRNA principles of action, learn molecular mechanisms that result in microRNA dysregulation, and identify inherited disorders caused by mutations in microRNA sequences. Sorry. So this is the first case presentation. It's two individuals from the same family, two brothers. The firstborn was a product of 37-week pregnancy. The pregnancy was complicated by polyhydramnios. The patient had duodenal atresia, TI fistula, small BSD and ASD. At physical exam, it was noted that the patient had microcephaly, small oral commissure, and it had, and it had the fifth finger clinodactyly with creases consistent with only 2 phalanges in each fifth finger. And the brother was born later. It was a premature baby, was born at 34 weeks, and it also had a superficial atresia, small ASD, also presented with microcephaly. And it was also noted that he had short palpebral fissures, micronasia, and there was no mention of digital anomalies. Oh, sorry, I was pressing, okay. So this is the family history. As you can see, the two brothers. There's a mention that the father had a swallowing problem and he had to have some endoscopic procedures. The paternal father had congenital heart disease and kidney failure due to recurrent infection. And then these brothers, they also had a cousin that had seizures in childhood, but not too many significant family history for this family. So in the differential for these two boys, there was mentioned that elements of this phenotype are consistent with the bacterial association. So this is typically defined by the presence of at least 3 of these malformations, congenital malformations. So vertebral defects, anal atresia, cardiac malformations, tracheoesophageal fistula with esophageal atresia, renal abnormalities, and limb abnormalities. So as you can see, this family presented mainly with 2 of these conditions. So there could have been another syndrome in their differential, and this syndrome is Feingold syndrome. It's a very rare syndrome, and it actually has 2 different types. And patients with this presentation, they have digital anomalies, mainly brachymesophalangy, toe syndactyly, and some hypoplasia is less common. They also have microcephaly. They have a mild learning deficit, short stature, and then they have other abnormalities, renal, cardiac. Some of them have hearing loss. And these features here, they are common both types, but then these other ones are more specifically for the type 1. So the type 1, they present with short palpebral fissures, micronesia, and then the GI atresias. So again, here I just detailed the different clinical features that these 2 boys presented. And as you can see, they presented a significant amount of them for Feingold syndrome. So Feingold syndrome type 1 is caused by germline mutations in the miRNA gene. It's a very rare disease. miRNA gene is located in chromosome 2 and is a member of the MYC family of proto-oncogenes that act as transcription factors. And the 2 proteins, MYC and miRNA, they show high homology, but both proteins are expressed in different human tissues and in different developmental stages. But they also, this gene is also somatically amplified in different types of cancers, which leads to overexpression and poor prognosis. But obviously, this mechanism is quite different than what the Feingold syndrome patients have. So here I'm just like showing you the first family that was originally described for this syndrome. When they found, actually, the first mutation that they found, it was actually a microdeletion of the area. And here you can see they did, you can see that the affecting members in black in comparison to the non-affected members in white, they have a weaker band because they actually deleted one of the alleles. And here you can see how they found it. It was done by mapping using microsatellites. So after they found this and they saw that they were segregating with disease in the family, they also had another number of individuals, other family, and they found also 12 different heterozygous mutations in the miRNA gene. So the main mechanism of function of disease in this gene is loss of function. And as you can see here, this is from ClinVar. The mutations are always located in one of these 2 exons. And the truncating variants, but also the missense variants are the ones that are pathogenic is because they affect crucial DNA bindings, because as I mentioned, this is a transcription factor. And also 10% of these individuals, they have a deletion of the short arm of the chromosome 2. And here I just put like the 23 pathogenic or likely pathogenic variants in ClinVar. So then Feingold syndrome type 2, it's caused also for germline deletion, but from the chromosomal region in the 13q31.3. And this region contains miRNA 17HG cluster. So this is also a very rare condition. And this is the microRNA cluster. So this it's the microRNA 1792 cluster in chromosome 13 that has these microRNAs in it. But also these microRNAs, they have 2 paralogs. One is in chromosome X and another one is in chromosome 7, and they share the similar sequences. And these actually these microRNA 1792, they have been shown to be implicated in significant cellular processes, and they are in controlling apoptosis, proliferation, and neuronal differentiation. So again, this is the original paper that describes the mutation in this cluster. They studied the 10 patients that they had clinical diagnosis for Feingold syndrome, but they could not find mutations in the and the MYC and locus. So what they decided to do, it was SNP array, and they found this mutation, one family that had this bigger mutation and this one smaller, and they saw that they were containing the microRNA. So here they compare that the affected members, they all had the deletion and the non-affected didn't have it. And because they found that they was affecting the microRNA cluster, they extracted RNA from lymphocytes, and they compared the expression of the microRNAs compared to controls. And here you can see that all the affected members, they had the lower expression. So what are microRNAs? MicroRNAs are a major class of small non-coding RNAs found in animals, plants, and some viruses, which regulate gene expression at the mRNA level. They function as they're like small guides of more or less 25 nucleotides that regulates the expression of mRNAs containing complementary sequences. And the first microRNA in humans was LED7. It was the first one to be discovered in 2000. And right now there are about 49,000 human mature microRNAs annotated in the miR-based database. So obviously, long way has been like an expanding field. So here I'm going to explain you just the very basic of the canonical microRNA biogenesis for you to understand how they are created. So most of the mature microRNA sequences are located in introns or exons of non-coding RNAs, as well as introns of pre-mRNAs. So the first thing that happens is that the RNA polymerase II transcribes these microRNA genes into large primary microRNAs called pre-microRNAs. Then these pre-microRNAs are cleaved into stem loops into the precursor called pre-microRNAs by the Drosha complex, which includes two main proteins, the Drosha and the DGCR8. And then these loops, they are exported to the cytoplasm by specifically the exportin 5, and then they are cleaved into near the loop into small double-stranded RNAs by the dicer complex. And finally, the microRNA duplex is then loaded into the Argonaute protein, which promotes the assembly of a ribonucleoprotein complex referred as the RISC by the RNA-induced silencing complex. And this is where it also binds to the target mRNAs. So here I explained you that explanation was about the canonical pathways, but also for you to know there are other non-canonical microRNA biogenesis pathways that are independent of the Drosha complex, as you can see here. And these are called miRTrons. So these miRTrons are also intronic regions that are processed by the spliceosome, as you can see here, and then they are debranched by the Lariat debranching enzyme into a loop of pre-microRNAs that they can again be exported. But obviously this increases the complexity of the biogenesis of microRNAs. So also, as I mentioned, one of the characteristics of microRNAs is that they can be placed located in the genome in clusters, as the one that I mentioned for fungal, or they can be single. So one of the important things is that you can see here, even though these are like microRNAs, because they're so important, these intronic clusters are usually highly conserved between species. And there are about 159 of these clusters reported in the human genome. So this is the main mechanism of action. The key point of the mechanism of microRNAs action is the seed binding. And that's a simple concept. I'm explaining a simple concept, but it's a more complicated. But here you can see this is the Argonaute protein. And then here you can see in red is the microRNA. And this about 8 of the nucleotides of the microRNA from the 25, it's what's known as the seed. And then it binds here with the mRNA. And obviously, this has different points of binding and interactions with the proteins in the complex. So, so that's the complexity and the different complementary that these small microRNAs have with the other members of the RISC complex. So also for you to understand in a very simplistic way, If the microRNA binds perfectly to the mRNA, then there is degradation. And if there is imperfect binding, then there is translational inhibition. And that's just like a simple way of remember the action. So now I'm going to go back to Feingold, now that we understand a little bit more about microRNAs and their action. And I'm going to explain you the scientific evidence that actually make them to show that they were implicated in this syndrome. So first of all, it was known before 2018, when they reported this paper, that the MYC family actually binds to the cluster to control the expression. So what this team they did, they created transgenic mice with different knockout models to actually see what was the effect of knocking out. So they had, these were the main constructs. They had mice that had a deletion of the MYC-N, then another one that had a deletion of the 1792. And then, as I mentioned, this is the paralog from the same family. So they did a double knockout. So here you can see they did the transgenic mice, and this is the control, and then they compare with all the other transcripts. So you can see here is that this one, that the deletion of the MYC-N in developing limbs resulted in severe and very severe brachysyndactyly, similar to what happens in Feingold syndrome 1. And then here is you compare, that's what we were mentioning before. This is the double knockout. So they also show that, and this is the single knockout, just knocking out the microRNA 1792. And you can see that it's not so severe, but it's also shortening the phalanges. And here you can see when they knock out both the two paralogs, they have also microcephaly, and they also see the syndactyly and the brachydactyly. So that's what resembles the Feingold syndrome 2. Then, because they knew that there was this interaction of the MYC-N with the microRNAs, they wanted to see what was happening actually. So what they did, they tested the expression of the mice, of the tissue, and then over time. So what you see is that in these mice that they have the knockout of the MYC-N, over time, all the 1792 microRNAs go down. So then what they did, they this is a control, and this is this mouse model that has the knockout. What they did, they took they overexpress the microRNA cluster, and they were able to rescue the skeletal anomalies. So that was showing that they are both implicated in these deformities, the microRNA and the MYC-N. So obviously, this is the summary, that the regulation of both genes are observed in are responsible for the skeletal anomalies. But I didn't show you all the extensive experiments that they did, but they obviously they were analyzing the different mRNA targets that they were differently expressed, and they ended up showing in the MYC mutation, downregulation of the phosphoinositol 3 kinase pathway. It's what actually is driving the skeletal malformations. And they actually proposed that there is a cross regulation of those. So in fact, in this syndrome, it's not that it's different, it's like they are both involved in the skeletal anomalies. So now that I explained you about Feingold syndrome, I'm gonna just explain you briefly and introduce these other diseases. There are not too many of them that are also caused by germline variants in microRNA sequences. So I will go over a very rare dysplasia, an ocular disease, and a form of deafness. So here I'm presenting these are the original families. There are not too many families that have this disease. This disease, it's a spondyloepiphyseal dysplasia, and it's known. It's characterized by a disproportionate short stature with short limbs, small hands and feet, and mid-face hypoplasia with small nose, as you can see here. This is the picture of one of the affected members in the family when he was young. And then the radiological hallmarks are the mild spondylar dysplasia, delayed epiphyseal ossification of the hip and the knee, and severe brachydactyly with cone-shaped phalangeal epiphysis. So these were the two families that they described, where they described this rare dysplasia. So because what they did, there was only 3 affected members. They actually did whole genome sequencing, and they found the same mutation in the seed sequence of the microRNA 140. So obviously, after they did that, they wanted to show that actually the mutation was affecting this phenotype. So again, so what they did, they created a transgenic mice, and they created one that had the homozygous mutation and the heterozygous mutation in the microRNA, and then they also knock out the microRNA completely. So here you can see that with the homozygous, they actually see that the mice were smaller. And then what's more characteristic of the clinical phenotype was this delayed ossification. So they were able to see that in the heterozygous and the homozygous, as you can see here, the heterozygous and homozygous, that there was this delayed ossification. And what's more interesting is that when they knock out the gene, the microRNA, like here, they had normal ossification. So, that was their way of showing that actually this was the cause of this dysplasia. So then I'm going to explain you about EDIC syndrome, which EDIC stands for endothelial dystrophy, iris hypoplasia, congenital cataract, and stromal thinning syndrome. And it's a local disease. And here are the characteristics of the patients. And then when they originally described this syndrome in 2002, they did a linkage analysis in this family that you see that there's several generations and affected members, and they were able to map using microsatellite testing that the genetic cause was associated with chromosome 15. And then 5 years, some years after, when they were able to sequence the same family, here you can see that they found mutation in the microRNA, in the seed sequence of microRNA 184. And here you see the chromosomal region with the microRNA. They show that this mutation co-segregated with disease. And here you can see how this variant, this nucleotide is highly conserved in different species. So I was trying to find an actual evidence of the implication of this microRNA mutation with the ocular disease, and this is the only paper that I have been able to find, because the original team, they didn't do actual functional studies. So what it's known is that this microRNA is the most abundantly expressed microRNA in the cornea and the lens. So this team, what they did, they actually used human lens epithelial cells, and they transfected a vector with the mutated microRNA that had the same mutation as the EDIC syndrome patients, and then the wild type. And then they compared the expression, the regulation of the expression. So one thing that they found is that the mutant one had a broader spectrum of genes controlling a broader spectrum than the wild type. And actually, they identified these 2 targets that were then regulated in the mutated in comparison to the wild type. So this is a mitochondrial dehydrogenase, and this is the receptor of a GABA-A. So this paper was more like creating more hypotheses on what it could be happening in this condition, because they were not able to find something very concrete. But they are suggesting that there could be some involvement of the citrate cycle, that because both of these, the GABA-A and the dehydrogenase, they are involved in the alanine, aspartate, and glutamate metabolism. So they just let it open, and I have not been able to find any other evidence of what's happening with this microRNA. So then I will explain you about a form, an autosomal dominant progressive form of non-syndromic hearing loss with postlingual onset. And again, this study also, when they described the disease, they also did linkage analysis, and they pinpoint the genetic cause to the chromosome 7. And again, similar to the other, when they were able to actually do sequencing, this is the very large family that they first studied with a lot of families affected, and they found a mutation in the miRNA 96. And then they had other families, and they were able to find another family that had a mutation in the +14. So the original one was in the +13. So what they did, in this case, they use bioinformatic tools to identify potential direct targets of the miRNA 96, and they assess the gene silencing capacity of both mutants using luciferase assays. So here you can see these are the predicted targets of the miRNA. So in black, you have the control vector, and here in red, you have the wild type. So they do the luciferase with the wild type, and they see that for all the targets, you can see the expression of the target, it's about 50%. And then they compare that with the two mutations in blue and in green. And you can see that when there is the mutation for all the targets, the repression is it's less. So that means that it's not having the effect. And what they are suggesting is that these targets they are important because in fact they are highly expressed in the inner tissue. So that was their way of showing that this has an implication. So interestingly, in the same journal, in the same Nature Genetics issue, there was this other paper that actually it's from the same authors, but they were able to just publish two different papers. So what they did in this other one, they described the same concept that miRNA 96 mutations cause the deaf phenotypes, but they did in another way. So they were creating mutants using this chemical compound, ethyl nitrosourea, and then they got one. They developed one after the effect of the chemical that was deaf, that they didn't hear. So they had two different types. They had the heterozygous, that they were showing a progressive loss of the prayer reflex, which is the response to sound when the ear flick responds to when they hear sound. And then they had the homozygous, which these mice, they never developed this reflex. And they also show head bobbing and stuttering and circling gait. So here I'm showing you they did scanning electromicrography pictures from the inner ear. And here you can see the significant difference between, this is the wild type, this is the structure of the inner ear, and then this is the heterozygous. And again, this is the wild type and the heterozygous, and that's the different architecture between the heterozygous and the homozygous. So obviously a significant difference, just like with the mutation of this miRNA. So to know what was happening in these mice, they did again mapping with trying to using microsatellites, and they map and they did sequencing, and they actually identified the mutation in miRNA 96. In fact, miRNA 96 is also part of one cluster with these other two miRNAs, and they show that the three of them are expressed in all sensory hair cells of the inner ear and in neurons of the cochlear and vestibular ganglia of newborn mice. And here you can see this is the SIT sequence of the miRNAs of this family, and this is the mutation, and you can see how this nucleotide is significant. Well, it's exactly the same in all the species. And here you see the heterozygous and the homozygous model, well, the DNA from the mice. So then what they did, they extracted RNA of the cells of the inner ear, and then they identified targets. And then here you can see these are the immunohistochemistry images of the wild type and the homozygous. And then you can see the difference on expression between the normal wild type and the heterozygous, and these are the homozygous. So what they are suggesting in the paper is that actually these three ones, they could well be involved in this development of the disease, because when you do a knockout of these single genes in mice models, they also develop deafness. So they were suggesting that any of those could be involved in developing hearing loss. So as a summary, I went over and I explained you about Feingold syndrome, that it's called for mutations of miRNA 1792. I also explained you that mutations in miRNA 140, it caused this very rare dysplasia phenotype. Then mutations in miRNA 184 cause EDIT syndrome. And then mutations in miRNA 96 cause a form of deafness. So now I wanted to explain you a little bit another type of genetic conditions that can arise from mutations not in specific the microRNA, but in factors that participate in the biogenesis of these microRNAs. So if you remember, I explained you that there's an important complex that is called the Drosha complex that has the Drosha protein and the DGCR8. Well, this protein is in fact the DGOR's critical region 8 gene. So here is the 22q11.2 region that causes DGORG, the deletion of it. And usually these patients have about 3 megabase. So when there is this deletion, what happens is that there is a haploinsufficiency of about 106 genes, which includes coding RNAs, non-coding RNAs, and pseudogenes. So here you have the protein that is here. And then also in orange here, you can see that there are also microRNA sequences. So obviously, there are other genes that have been associated with the phenotype of DGORG. But what I'm going to try to explain is that because this deletion, it has an effect on this protein and the other microRNAs, then the deficiency of this could have an implication on the complex phenotype of this syndrome. So because this protein is essential in the biogenesis, if you have a deletion of one of the alleles, like obviously the theory is that you are gonna reduce the expression of a lot of microRNAs because they cannot be processed. So consistent with this, patients that have a deletion in this region, they actually have a microRNA dysregulation in lymphocytes when they extract the RNA with a significant reduction in comparison to controls. In the mouse models, haploinsufficiency of this protein causes about 50% reduction in the overall expression of microRNAs in neurons, and that the neurological microRNA deficiencies that are observed in the heterozygous mice model for this protein are very similar to the same models that have a deletion in mouse. So it suggests that the reduction of the expression of this protein is key in those microRNA deficiencies. So again, it's not just the haploinsufficiency of that protein, but also there are like 7 microRNAs in this region. And microRNA 185, it suggested that it has the strongest connection with the actual clinical phenotypes. And patients with the deletion syndrome, they actually have a lower significant expression of this microRNA in blood samples in comparison to normal individuals. And this microRNA is expressed in cardiac tissue, immune cells, and in the brain. And they suggest that this microRNA is specific because the deletion disrupts key pathways that are connected to the function of this microRNA, and that these pathways are related to the clinical phenotypes of the patients with the deletion, including abnormal neurotransmitter release, low T cell output, cardiac hypertrophy, and autoimmunity. So now going back to the original case presentation, this is actually the mutation that we found in the 2 brothers. we found a miRNA frameshift mutation that it was novel. It was not reported. It's not reported in gene data in the genome, and it's de novo. We didn't see that it was reported in the literature either. And this is to go back to the pedigree. We tested the parents, and we were able to see that this mutation was paternally inherited. And as I mentioned before, the only note that we have about the father is that it has this swallowing problem, but there is not much more information about this family. So I wanted also to share with you an update on these two brothers. So the update that I have for the first brother, it was from the notes when he was 3 years old. The patient obviously had several surgical procedures to help with the repairing the atresia, and it had a gastrostomy tube placed. These are the observations and reported by the mother of these kids. The mom said that the patient never really smiled when he was a baby, and that he has had significant developmental delays, that he has low tone, he still has trouble swallowing, and he had strabismus, and they was given a patch, but then now it's only mild. And for the other brother, the younger one, at 5 months old, the mom says that the patient smiles more and has a higher muscle tone than his brother, that he's still learning how to swallow and has issues with emesis and gagging. He also has a J and a G tube, and he was given a Prevacid, but that he's gaining weight rapidly. That he's still not rolling over, but he's starting to turn over on his side, that the microcephaly is improving, and that he had a kidney ultrasound that was normal, but they were noting that the right kidney was slightly dilated. So with that, I also wanted to provide a clinical criteria so that it could be used in the clinic to identify these patients if we ever have another one at Yale. So this group, they analyzed all the more than 120 cases that have been reported in the literature, and they also added additional 11 cases. So they suggested that we could propose to have one set of major criteria and one set of minor criteria. So if at least 3 of the major criteria were fulfilled, or if 2 major criteria and 3 minor criteria were fulfilled, then there could be enough suspicion for to think that this patient had clinical Fengel 1 or 2. So the major criteria include microcephaly, GI atresia, a loss of function variant in the MIG engine, brachymesophalange, and a family positive history of the syndrome. And then the minor criteria include short palpebral fissures, short stature, syndactyly of toes, mild ID, and hypoplastic thumbs. So as here, if the criteria are fulfilled, then MICAN analysis should be done. And then if it's negative and the patient doesn't have GI atresia, then consider for testing for the miRNA. But then they also wanted to make a point that if there is severe ID or more of a complex phenotype, that consider other diagnoses. Also, even though they could find even a mutation in the gene. So with that, this is the take-home messages that I hope that I have been able to give you. So miRNAs are key regulators of the human physiology and create complex regulatory networks. The genetic disorders caused by mutations in miRNAs are very rare, but they exist. And that's my personal kind of point of view. The last one is that the new technologies such as long-read sequencing and whole genome sequencing will allow for identification of more of these type of diseases. Because as if you've been following my talk, I've been trying to explain you what was the technique that they used, molecular technique, to identify it. And just one of the, for one of the diseases, they use whole genome because they only had like 3 affected individuals. But for all the other ones, they use old school linkage analysis because obviously we've been in the area of exome sequencing. So obviously we have not been sequencing the introns. So I really think that we are gonna, because of the big regulatory effect that microRNAs have, and now that we are gonna start heading for more intronic sequencing with the whole genome and long reads, I think that we are going to start having more of this disease described. So with that, I just would like to thank everyone that it's part of my LGG training, and just say thank you for listening, and I'll be happy to try to answer any questions you might have or have a chat about any comments. Thank you.
So, Rosa, I don't know if we're picking up microRNAs or not. Have you looked? I mean, they may be in our exome sequence because the exome it's twice as big as the coding sequences of genes. So it has a lot of stuff in it.
Yeah, I have not looked. I've been thinking about this, but I have not looked at it. And I know that also with the exome, sometimes we just also have like deep coverage for regions that are in the intron. So I'm not sure if, if it could be something that we can look, but obviously the exome is not designed for like identifying like targeting sequence of the microRNAs. But now that we are going to kind of move towards, I think that that this is going to be a big thing to kind of start getting these sequences and being able to identify these mutations. So we'll see. I will take a look and see if we can find any.
Yeah, I don't know. I mean, and we can always add things, but we can't add all of the microRNAs. It's probably too much, but we can always add things to our capture set. I just don't know. There are other things that we used to miss that were like this, like telomere RNAs used to capture them.
Yeah, and obviously all these conditions, they're very rare. Like some of these conditions that I explained, there's been like just like a handful of families. Like for Feingold one, no, that one is more common, but all the other ones are very, very rare. So yep. I don't know. So there's Mohammed, I think you have a question. And
yes, my understanding for microRNA, they use the Tachman probe, the PCR, to pick up this microRNA, and then they do reverse sequencing. So the technology you're talking about, they are doing sequencing on the whole genome and whole exome?
No, how the original old school was doing linkage analysis using microsatellites. So like that, you are only able to pinpoint like a chromosomal region, okay? And then all these experiments, they were doing that. And then when they had access to sequence, they just like sequence the actual chromosomal region. But then with long reads and like whole genome that it's based on long reads, now we can start like sequencing like the whole intron. So now we are going to be able to be sequencing, because with the exome is like captured for the exons, and microRNAs are not in exons, they are in introns. So we have not been sequencing them. Do that make sense?
Okay.
Yeah. Yes. Okay.
Yeah, Dr. Xicola, I want to know if in your search, whether you came across miRNAs that have been shown to be associated with very early pregnancy wastage. Because the phenotypes that you have presented, it looks like a lot of them are later in the developmental process.
So you say if I have come up with diseases that are more in kids,
for example, just embryo lethality.
Oh.
Or
okay.
Early pregnancy loss.
Yeah. So I have not. So I was trying to focus on diseases that have an OMIM accepted, even if it's very rare, that at least has an entry in OMIM but I have not found on what you are mentioning, like pregnancy loss or stuff. So I'm sorry, but I didn't. I actually didn't come up with any. I don't know if there's some research, but I tried to focus it only on like established OMIM diseases. So if no one else have any questions, just If you need the code for the CME, it's in the chat.
Oh, hi,
Yes.
Rosa. I actually put a question in the chat.
Okay. How do you see long read sequencing adding value besides possibly facing the compound that out? Because you know, if the long reads you can like decide to do like the whole gene in a more efficient way. That's why I meant.
So similar to like whole genome sequencing.
Yeah.
Okay.
But-
Okay.
Yeah, so that would be, so, you know, imagine that, for example, you have some evidence of a microRNA cluster, right? So you
Mhm.
would be able to kind of do it more efficiently or than if we would not have it. So in that sense, that we are going to be able, the long read, because we can just kind of sequence like bigger chunks of the DNA. That's what I
Mhm.
meant. But yeah, it's
Okay.
similar to the whole genome.
Okay. Yeah. Thanks. And I think Hui was talking, but we couldn't hear her. I think she had something to say too.
We cannot hear you. Wait, we cannot hear you.
Hi, Rosa. Can I ask a question?
Sure.
Have you encountered anything kind of related to like a dosage effect from those microRNA? Because from some of the cancer cases, actually, we see deletions. The impact actually is caused by that dosage effect from the microRNA.
Can you repeat? Pinging.
Have you noticed any dosage effect from those microRNA? That means like if you have a deletion or duplication, they can affect the downstream and then
Yeah,
cause
that's
some.
so that's what happens with the fungal. There is the deletion of the chromosome 13, that area, and then you lose one copy of all these microRNAs, and then that's the effects that they were showing. So that it's actually, obviously it's all the genes that are regulated by the microRNAs, right? That are having developing the phenotypes. But that's what it's more, some of these diseases that I was explaining are more point mutations on the seed sequence, but then also the other ones are like the deletion of the whole sequence of the microRNA. So yeah.
Yeah, this is somehow related to the previous question about the lethality in you know, some of the spontaneous abortion, we know like 50% of those are caused by aneuploidy. Those are kind of have either, you know, rarely they have monosomy, but mostly have trisomy of some chromosomes. So that means if you increase the dosage of microRNA across the chromosome, they may have some downstream impact on the fetal development, and then that could cause some lethal conditions.
Yeah, obviously
Yeah,
that's the other one. I didn't come up with any on my literature search on like what you are saying, but obviously like overexpression of microRNAs also like it's gonna have an effect. But none of these conditions they were well, the other overexpression, it was when I was explaining that they were able to rescue actually the skeletal anomalies for the fungal, but obviously that's the other part of that could happen. If you have a duplication, then you are overexpressing the microRNAs, and that is going to have an effect. So I think that the way you wanted to, do you mind reviewing the slide of the connection of the MYC and the microRNA? Oh, okay. Sorry. Oh, I've started. So here, that's what you mean. So let me see. Wait, can you say if this is what you want? Sorry. Okay, I think that's what it is. So the connection is that they actually go through different pathways, but the MYC-N is actually regulating the expression of the microRNAs. So when there is the deletion of the microRNAs, but they did a lot of different experiments, and I didn't put all of them here, but they see that there is an upregulation of the TGF-beta, and that's what's causing the skeletal defect. It's driving the skeletal defect in fungal type 2, but in the MYC and deletion, there is the main driver if it's the downregulation of this other pathway, but that they are actually influencing both. So it's not that they are two different diseases because it's caused by deletion on these two genes, is that they are affecting both mechanisms. So that's it was trying to give a summary for not like overcrowding the the talk with a lot of mice models. So I'm gonna stop sharing and see if that's what you wanted. Wait. Okay.
Yeah, I actually find that a little confusing because the mouse models really make it look like the effects of loss of MECN are entirely mediated, or large, at least on the skeletal thing, mediated by the microRNA. You know, because you can rescue those effects by just overexpressing that microRNA. So it seems sort of conflicting to think that they're using different pathways, because it just feels like the microRNA is simply downstream from MYC.
Yeah, but it's, I think that what they were trying to say is that there are these other mechanisms involved that are not exclusively the deletion. So because obviously in the microRNAs, they are affecting different targets. So I think that they were trying to tease out a little bit more and then just not say that it was just the deletion per se.
Yeah.
So
Okay.
that's what they were trying to say.
Yep.
So I think that if no one else has any questions, just gonna go with your day. Thank you all for listening. Bye.
Thank you.
Bye.
Bye.
Thank you, Rob.